Hello, and thank you for joining this presentation, The Biochemistry of a Dark and Stormy and Other Ferments. I'm Laurel Westendorf, part of the Community Relations team at the Deschutes Public Library. Every month we explore a theme, and this month we're exploring fermentation. Our presenter is Jeannie Garbarino. She is a scientist and educator at the Rockefeller University in New York City, where she serves as director of RockEDU Science Outreach. Thank you so much, Jeannie, for helping us understand how fermentation works in such a fun way through a cocktail.
Thanks for having me. I'm really excited to be able to talk to folks on the West Coast. I'm very used to talking to New York City communities, and I think one of the wonderful benefits of our pandemic context is breaking out of our little bubbles and starting to interact with people outside of our own spaces. And so again, thank you for having me. I have the privilege of serving as the director of RockEDU Science Outreach at the Rockefeller University, which is a phenomenal biomedical research institute on the Upper East Side of Manhattan in New York City. And through this work, we aim to connect non-scientific communities with the research process with the hope of fostering meaningful connections with scientists and to foster an appreciation for science as a human endeavor. And we do that a lot through topics that I feel are really accessible. And one of the most exciting things that I find, and the most exciting research topics that I find, is fermentation. It's my favorite scientific topic, and it's also my favorite hobby at home. And more specifically, I love the Dark and Stormy cocktail. It's my favorite. And one day as I was sipping it, I was thinking, wow, there's a lot of science going on in here. I should build a talk around it. And so I want to talk with everybody here about how we as humans have been exploiting microbial ecosystems for generations and how this process really helped to give birth to modern science. Okay, I think it's time for us to get started. I'm Jeannie Garbarino. I'm excited to talk to you about the biochemistry of a dark and stormy and other ferments. I want to first start off by talking to you all about microbes. So what actually is a microbe? I know this feels really scary. And when we think microbe, maybe we're thinking of SARS-CoV-2, or maybe we're thinking of that stuff that you see in the back of your throat when you have strep throat. But really, microbe is any living organism that is too small to see with the naked eye. It can consist of a single cell, which would be unicellular, or it may be made up of several cells living in collaboration with each other, which would be termed multicellular. And the cool thing about microbes is that they are found everywhere, and I mean everywhere. You can find them on every surface, on every piece of your body, in the dirt. I think a teaspoon of seawater has something like 12 million virus particles inside of it, among other types of microbial particles. And they play really important roles. And that's because microbes that are naturally occurring, not the microbes that we try to culture in labs, microbes have always lived in something called symbiosis. And symbiosis basically means that it is a close association between two or more organisms of a different species. And so what does that mean? It means that, we have bacteria, for example, in our guts that help to make sure that we humans have vitamin K, as an example, right? And so microbes can exist in symbiosis with other types of hosts through a variety of different mechanisms that span from something called commensalism. This is where you get a benefit from one, the benefit goes to one of the organisms, but nothing really happens to the other organism. So it's like neutral. You can have mutualism when you give a benefit to both the microbe and the host. And then you can have parasitism, which is what's taking place right now with the SARS-CoV-2 and COVID-19, is when one symbiont, such as the virus, benefits at the expense of another symbiont, which is for us humans. And as I had mentioned, you exist along the spectrum of parasitism, commensalism, and mutualism. And when you're more parasitic, it's antagonistic to the host, so it makes the hosts very sick. SARS-CoV-2 as an example. I'm not going to stop. That's the easiest example for me. Or you can have something that's mutual where there's a lot of cooperation taking place. And it doesn't always have to be, this whole thing about Antagonism and cooperation, or parasitism and mutualism, is not just with microbes. It could happen with any kinds of organisms, like honeybees and flowers, as an example. All right, so how does this all play into fermentation? I really like fermentation because it showcases how microbial ecosystems are really, really at play. So food fermentation is made possible through the natural microbiome that actually exists on all food. So as I had mentioned, microbes are everywhere. So you have common food products, things that are fermented a lot, like cabbages or beets, carrots. I love fermented carrots. You can make kimchi and all sorts of pickles, right? But really what this looks like when you put it under a microscope is this. It's a fruit salad or a food salad of all sorts of microbial life. And so the idea is to exploit that microbial life that's naturally existing on the surface of these vegetables to make delicious culinary foods. So fermentation is really key in that it helped to propel the birth of modern biochemistry and microbiology. So from a culinary standpoint, however, just going back, fermentation really exists as the transformation and preservation of food by microbial organisms. And as I had mentioned, we have been doing this for hundreds of generations. And this is because we didn't have refrigeration and people wanted to preserve food. And this is the way that people did it. And our first elements of, or first evidence of fermentation really dates back to China in 7000 BC. And on the left, you'll see an image of pottery. And anthropologists have done some chemical analyses of the surface, of the inside surfaces of these pottery to find, the types of chemicals that existed, and they've traced it back to fermented foods. And they invented a beer type of beverage called kui, made with rice, honey, and the fruits of grape and hawthorn plants. And so this was the first, one of the first evidence that fermentation dates back that long ago. And of course, it has to deal with, alcoholic beverages. I feel like a lot of what we do and what we know is really spurred by alcoholic beverages. And later on, the Babylonians in about 4500 BC is when we found the first evidence of beer. And we found the recipe for beers on clay tablets. And this is like the wild beers. So this wouldn't necessarily be the beer that like Sam Adams has engineered yeast that they use and it's like consistent every time. This is very much like what is around, how, the sort of lacto ferment type of beer that has that tangy kind of flavor. And one of the facts that I really appreciate about the ancient Babylonian beer making situation is that the majority of the brewers, actually all of the brewers, were women. And this was one of the professions that was highly respected, like a lawyer or doctor. And they even had their own divine goddess to protect the brewers and the brew. So I thought that was very interesting, which is much different from a lot of what we're seeing with these microbreweries and who's running them now. So we'll fast forward a tiny bit and we can see the Egyptian civilization was among the first to use yeast to leaven their flour to make bread, also to brew beer, which was actually one of the most abundant components of their diet. So this is again at a time where there wasn't a lot of plumbing or hygiene with water and things like that. So to drink a fermented beverage was actually safer than drinking water. They also fermented dairy products and were among the first civilizations to produce cheese and yogurt. And of course, that delicious food that we all love is butter. So moving along the culinary fermentation timeline, we see that a little bit later on, the ancient Babylonians were able to preserve meat sausages. So they made things like whatever the Babylonian equivalent would be to like salami. Fast forward a little bit more and we see widespread fermentation of vegetables and legumes taking place in China. A little bit further along, we see fermentation of sweet tea. So if you all are drinking kombucha, that probably originated in both China and then Japan, with kombucha emanating from the Japanese culture. And between 500 and 1000 AD, we see almost global cereal legume fermentation taking place, like miso and dosa and sake, which are staples of many, many different cultures, pun intended. But really, it wasn't until sort of this, you know, the sort of privileged few that got to mess around in laboratories in Europe and Western Europe that we started to develop an understanding of the fermentation process. And the first person to document a cell was this Dutch scientist, Antony van Leeuwenhoek, who he built the first microscope, and he looked at cellular material under the microscope and dubbed them as something called anomolecules, because he noticed that they were sort of living and metabolizing or like entities. And he was he documented this thing. Imagine being the first person to look at a cell. I am not sure. I think this probably would have been one of the most incredible moments across human history, is to see a cell for the first time. So he is dubbed the father of microbiology. So he really, suggested to the scientific community, which was very new at the time, and it was just really forming, that, hey, they're cells, and cells are associated in some way with life. Then came along a German scientist, Justin von Liebig, and when it came to fermented foods, Antony von Leeuwenhoek really was established that cells are things that make up living organisms. And we know that there's this process of fermentation that's been established for generations. And in the study of trying to figure out how fermentation actually takes place, von Liebig theorized that it was the death and decay of the microorganisms around the food that gave rise to the fermented sort of product of the food. So that was a little wrong, but that's okay, because in the 1800s, I think one of the most famous microbiologists emerged onto the scene, and this was Louis Pasteur from France. And interestingly, a little vignette. So there was I guess in France, they had a lot of beet juice fermentation. And there was a big beet juice fermenter processing plant that was run by this guy, Monsieur Bigot. And one day he came across his vats, and he saw one of the vats, was taking place normally, and it had the nice sort of ethanolic, alcoholic flavor taking place. But the other one just tasted sour, vinegary, in fact. And his son was hanging out in the laboratory of Louis Pasteur, and his son suggested that this brilliant scientist should come in and come check out the vats of the beet juice to figure out what exactly is the problem. And so when Louis Pasteur looked at the normal ferment of the beet juice, he saw these big fat globules in there, which we know right now is a microbe called Saccharomyces cerevisiae, which is something you are all familiar with. This is just budding yeast. It is the yeast that makes our beers and our breads. And in the sour vat of beet juice, he saw that the globules were more, they weren't as round and buxom as the normal ferments, but instead they were sort of long and cylindrical. And he explored it even further and figured out that there's all sorts of microbes that give rise to fermentation byproducts. And the fermentation byproduct that was produced by yeast is ethanol, whereas the fermentation byproduct that was in the sour vat was probably from a bacteria creating some sort of acidic material. So either lactic acid bacteria or acetic acid types of bacteria. And essentially through this quest to restore the alcoholic beverage field of beet juice, Louis Pasteur helped to create the field of modern microbiology. I think we have a theme here. Alcohol really drives science. So we've talked a little bit about the culinary definition of fermentation. We talked a little bit about the history of how we got to where we are, or, the path that brought us to where we are today. And so I want to talk a little bit about the scientific definition of fermentation. And this is a cellular process through which organic compounds are converted into energy. And that is the key. It is all about energy, right? And so when we just intersect the culinary definition with the scientific definition, what we have here is the birth of modern microbiology. And so what does that process look like, right? So anytime fermentation takes place, it always starts with a sugar. And this sugar, the most common sugar used in fermentation is one called glucose. Glucose is the common energy source of almost all forms of life. And it is utilized, it is sort of inserted into a cellular path called glycolysis, which I've indicated in blue script at the top of the page. And interestingly, glycolysis is actually my favorite metabolic pathway. It is conserved across all organisms. I mean, everything living and extinct has used glycolysis in the past. And the way it works is glucose is fed into a metabolic pathway and eventually produces something called pyruvic acid. And pyruvic acid is then fed into a fermentation system to create fermentation byproducts. But if you remember, I discussed with you before that it's really about, the scientific definition of fermentation is about the creation of energy, right? And so energy is really the key for this. And all of this other stuff, pyruvic acid and the fermentation products, it's just sort of byproducts. This is microbial waste, right? Because what the bacteria and other microbes are really interested in is this something called a redox reaction. So there's a special molecule called NAD+, and it becomes reduced into NADH, right? And then NADH is in turn oxidized to NAD+. And in order to turn this cycle, you have to take glucose and convert it into some sort of fermentation byproduct. And it is through this redox reaction that microbes produce energy that sustains life. So things like ethanol, lactic acid, propionic acid, the common elements and byproducts of fermentation are not actually useful, metabolically useful for the microbe, but instead are microbial waste products. And so in terms of the basic fermentation here, we know that microbes need to make energy to survive. They do so by using a carbon source, usually glucose, to generate energy, and they make these waste products, and we essentially love to eat microbial waste. And I think that's the coolest part, is like we are exploiting microbes, not necessarily for these like really important compounds, but really for their excretions. And so the microbial excretions are actually amazing and delicious. As I had mentioned to you, we have glucose that gets converted into pyruvate, and then pyruvate, depending on the organism, is converted into a number of different compounds. And so, for example, Aspergillus, which is this type of mold here, it creates lactic acid, and this is a common culture for the mass production of soy sauce. Lactobacillus, everybody is probably, if you've eaten yogurt, or if you've had a wild beer, or if you had a fermented vegetables, you are most certainly eating lactobacillus. And it's the lactobacillus produces something called lactic acid, and we find this in things like, again, cheese and yogurt and stuff like that. Then you have the yeast, which is Saccharomyces species, and Saccharomyces is responsible for the production of ethanol and carbon dioxide, and so that's what's really giving the rise to the beer. So this is the Saccharomyces, the bottom panel here, and then the middle panel is the Lactobacillus. And just to go back on my vignette with Louis Pasteur, the healthy vat of beet juice contains something that looked like this on the bottom, whereas the soured beet juice contains something that looked like this in the middle. And so that's how they knew there was something wrong. So Saccharomyces, going back to the yeast here, they produce ethanol and carbon dioxide, and those can be harnessed either together to produce beer, or separately to produce wine or bread. And it is the carbon dioxide released by the Saccharomyces that gives bread those like little holes and makes it fluffy. And if you've ever heard of people eating unleavened bread, you know, unleavened bread doesn't have that sort of fluffy nature. It's because it doesn't have yeast. All right, so where does the Dark and Stormy come into play? Interestingly, the Dark and Stormy is a patented and trademark beer. Patented and trademark cocktail by Gosling Brothers Limited. And the official recipe consists of 1.5 ounces of Gosling's Black Seal rum, topped with ginger beer, and it's served in a highball glass, and you may use lime if that floats your boat. And how did it get its name? Well, the color of a cloud that only a fool or a dead man would sail under. So an English sailor once said this, and they felt that the Dark and Stormy resembled that color. And so that's how it got its name, the Dark and Stormy. And so let's deconstruct the ingredients of the Dark and Stormy, now that we know a little bit about fermentation, right? So Gosling's rum is made with a combination of molasses. And so molasses is a carbon source, sugar, it's unprocessed, right? And it also has a little bit of white sugar. And the microbe that utilizes the sugar is yeast. And so you're getting that ethanol byproduct, that ethanol waste product from the yeast cells that sort of create the rum here. Now, ginger beer is made with ginger root. So this is something that you can buy in the supermarket, and it naturally contains a microbiome on top of it. That's mostly a lactic acid type of microbiome. It also has some naturally occurring yeast. And so you take the ginger root, right, and you combine it with some white sugar and water, and it has its own yeast and bacterial system already existing on it. And you put those together and you have the products of, these are all products of fermentation. And so again, just deconstructing this, you have carbon source, right? And you have microbes, and you make something delicious. Okay, so how do you ferment ginger root? Well, ginger is, as I had mentioned, naturally has a microbiome. And a microbiome is termed as the collection of microbial organisms living on, in, and around something. And the way I create a ginger bug is to grate the root using a microplane grater, just add a bunch of sugar, right? And it sort of has its own liquid, and you're letting it sit there for 3 or 4 days. And the microbes are using the sugar to create energy while also producing their waste products such as lactic acid and ethanol. So once I have that for 3 or 4 days and I have a rich microbial culture going, and then you're leaving this just on a shelf or at room temperature, right? And this is what my ginger bug looks like from a couple of years ago. And then you take this and you'll add it to a larger vat for making ginger beer. And people have been harnessing the natural microbes already existing on ginger root to start the brewing process for a very long time. And so here's the ginger beer recipe. So I start with 4 liters of water. I ground 6 inches of grated ginger. I add a cup of white sugar, and I have my ginger bug starter, which I had made previously. And I simmer the 2 liters of water. First, I simmer 2 liters of water with some ginger root for 15 minutes. Then I add some sugar. I let it cool to room temperature. You don't want to cook your I mean, this is the whole process of sterilization. You're heating something to kill the microbes. So you don't want to actually have heat because you want the microbes to live. So you let that cool to room temperature, you add your ginger bug starter, and you ferment for 24 to 48 hours. I suggest using a flexible container, not a glass container, because your glass container could explode. If you want it to be really, really bubbly, you can also add a little pinch of baker's yeast that you can just get at the supermarket, like Red Star or something like that, or Saf yeast. And then once you've fermented for 24 to 48 hours, pour it over ice, add your rum, add your lime, and there you are, the Dark and Stormy. All right, well, that is my talk on the biochemistry of a Dark and Stormy. So it's really a talk about fermentation. Hopefully the Dark and Stormy drew you in, but the fermentation science made you stay. Thank you for joining me. Again, my name is Jeannie Garbarino. I'm the Director of Rock EDU Science Outreach. I hope that you'll check us out, and I'm happy to take your questions. So thank you very much.
Thank you so much, Jeannie. I wouldn't have realized that you could make ginger beer. It wouldn't have never occurred to me that ginger beer's recipe would be that simple, really.
Oh yeah. Honestly, like I make a ton of ferments and so basically all you have to do is create an environment for it to outcompete the bad stuff, right? So most of the microbes that exist on our planet are actually either neutral, like have no impact on us. For example, we're at any given point in time, we're infected with 12 different species of viruses that probably do nothing to our bodies. And then you have the situation where they can be really helpful, like healthy guts are supposed to be linked to, and by healthy gut I mean a good diverse microflora that it lives in our guts. It's supposed to be really good for your health, really good for your mood. There's some suggestion, and I think that the jury's still out in terms of exactly how strong of a link this is, but there's suggestions that it can be related to keeping good heart health, keeping good mood, things like that. So there's a lot of really interesting research coming out right now about the microbiome and all sorts of human health and disease.
Well, I really appreciated your demystifying everything and being like, no, actually, this big long word, you know what this is. You've eaten this. That was very helpful. But I find it so fascinating that the things that you were listing that have been enjoyed by humans thousands of years ago, like the sweet tea, the miso, the salami, the butter, the cheese, we've been eating these same foods since the beginning of fermentation.
Absolutely. And this is necessity is the mother of invention, right? So how do you make sure that there's enough food for people? You get milk or you harvest some grains or vegetables, but this has to last some amount of time, and you don't have the capacity to preserve it through what we use now is refrigeration, right? There was no electricity up to about 150 years ago. So these are the processes that are absolutely standard across history. It's just to make sure people aren't wasting food.
Yeah. And it's a good thing. They're all delicious too. So.
Oh yeah. I mean, I ferment. Honestly, one of my favorite things is I'll just take carrots and I'll slice them into spears, and I'll just sort of grab like a mason jar and have them just below, fill it with water, add a tablespoon of Celtic sea salt. It's the sea salt that you can buy it on Amazon. It has like a blue bag of it. It's the most delicious. It's gray. You put it in the jar with the carrots for maybe a couple, some garlic, maybe a little thyme, something to spice it up. And it's honestly the best snack in the world. My kids eat it like crazy. This is how I get my kids to eat their vegetables, is fermenting them.
And they're probably, I don't know, maybe even better for them. So,
I mean, possibly, yeah. I mean, it certainly isn't candy. It's not crap,
What I've noticed is that people that are really into fermentation, like we're having Sandor Katz later.
Oh yeah. I met him, and I like totally fangirled over him, and I like have like a selfie with him, like as a fermentation like junkie. Like
right.
this is my guru, So
right.
Yeah.
And what I find so interesting is that those of you who do it so regularly, that ferment things all the time, are just like a little bit of this, a little bit of that.
Yeah.
And I'm kind of a recipe person. And what I've noticed is that when people get really comfortable, it's a lot more flexible. It's a lot more creative, what you have, what's delicious.
Yeah. Well, and to be honest with you, we really are trying to push that science is not just following a recipe. It's not just following a protocol. It's thinking, it's putting together things and watching and observing how it works. And then it's reflecting on that process and then trying to repeat it, maybe making some modifications. So it's this series of iteration and reflection and trying something over again. And things like fermentation are these really robust yet super flexible systems to really explore the process of science, and really which is defined as like iteration and reflection. And So we over the summer would make yogurt with students virtually and have them do all sorts of experiments with it as a mechanism for them to explore the process of science and also have some delicious snacks along the way.
Right. Well, I love it. I love the thinking beyond just a recipe. It's like,
Thank you. Yeah.
Well, this has been great. Well, one last question. Louis
Sure.
Pasteur, is that where pasteurization came from?
Yes. That is the namesake of pasteurization. So actually, Louis Pasteur gave us something I think that was it was really important for us to understand, but I think we kind of like ran away with it and to our detriment. And he gave us something called the germ theory of disease. So he was the one who was able to link these parasitic microbes with human disease. And I think for a very long time, human society was very scared of microbes as a result. And we wanted to sanitize. You know, back in the 90s, for sure, there was always like antibiotic soap and take all these antibiotics. And then we realized, wow, that is a terrible idea, right? Because we were really shifting that symbiotic component that we need. We're shifting the natural order of things by destroying specific microbes. And so while it was really important to recognize that microbes can cause disease, I think it was actually detrimental to our society in putting all of our eggs in that basket and not really understanding the benefits that microbes give us.
Right. It's not just,
and not to mention, exactly, not just Black Plague, not just SARS-CoV-2, but it also is, microbes are these really essential things for bioengineering. You know, a lot of the scents that you smell, like in your candles from, like, I don't know if you have like Yankee Candle Company out in Oregon or anything like that, but you know, but these are all generated usually through huge amounts of microbes. So they genetically engineer the microbe to produce this compound through fermentation. And then they isolate the compounds and put them into candles. And that's just one example of how that works. So
wow. So that pumpkin spice smell is really
probably made with yeast.
Great. Well, thank you so much, Jeannie. And I will make a Dark and Stormy now and just watch.
I hope you will.
Bubbling.
Yeah. I have to say, I also like a little bit of Schweppes ginger ale. It's fine when I'm feeling like less fancy. But right. if I'm ready to drink with Pinky up, I'll go get my ginger beer. You know.
Ale is just as good. Noted.
Yeah. Totally.
Well, we have a lot of other great library programming available for you in the audience, and it's all fun, free, and virtual. You can go to DeschutesLibrary.org and look at our calendar or our virtual event guide. So have a great day, everyone.
Thanks a million.